Okay, thank you all for joining us for this conference on a really important and exciting topic, I think, and that is the idea of using therapies for Duchenne in combination. It's really amazing that we can even talk about this topic because, 10 years ago, we were just excited about having any one single therapy. Now it looks like we might have multiple things on the horizon. And it's clear that this is a disease that will likely be best treated by combining different therapies. They may act on different targets. So what you're seeing on the screen right now is just a sort of general schematic that shows all of the different targets that have been identified. Of course, the loss of dystrophin is what is responsible for causing the symptoms of the disease. But downstream of the loss of dystrophin, a whole cascade of things happen in the cells, and these things all present targets. So I'm not going to go into detail about this chart. I just want to give you an overview. Dr. Lee Sweeney from the University of Pennsylvania and Dr. Dominic Wells from University College in London are going to give you sort of the highlights of their thoughts about combining therapeutics. And then afterwards, we will have time for questions. You can type your question into the chat box that you see. We'll see those questions, and anything we don't cover during this time, we will try to follow up with Lee and Nick afterwards and get you those answers. So we'll cover as much as we can during the presentation. So everyone on the call is muted, just reserve the sound quality. However, you can type in at any time during the presentation questions into the chat box, and we'll ask them at the end. So with no further ado, I'm going to turn over the conversation to Dr. Lee Sweeney.
Okay, thanks, Sharon. As Sharon said, I'm at the University of Pennsylvania, and a lot of what my lab does is really devoted to trying to find therapeutics that will change the course of Duchenne. So we've all been facing the current realities, the challenges that the dystrophin restoration therapies that have been in trial do appear to slow the disease, but they haven't halted it. I mean, because of this, there's been problems even with the early trials getting approval. As you know, the Autolearn 2B trial did not get approval because of an unexpected dosing problem. The GSK phase 3 trial failed to get approval, even though it really looks like in analyzing the data that at least subsets of the patients receive benefit. So I think it's true. I think we're going to see, as we have now seen in Europe with at least conditional approval of Autolearn, that these dystrophin restoration therapies are going to probably be approved in this patient population. But the reality is that they're not going to be a monotherapy. We need more than these because they just aren't enough. And besides, they're only applicable to a subset of the patients at this point. So there are many patients that are not eligible for these treatments at all. So the opportunity, though, is, as Sharon said, there's a lot of facets of this disease that are downstream of the genetic problem, the fact that you're missing dystrophin. There are lots of other things you can go after, and I'm just going to highlight a few, but you can think of them as lumped into sort of these 5 categories, and 4 of them have nothing to do with dystrophin. You could try to do a better job of driving the muscle regeneration and even improving the mass and strength of the muscle. You can, inflammation and fibrosis are really the problem that interferes with regeneration in the muscle. And so if you go after that, you could improve even without anything that builds muscle mass. There's a blood flow problem in the skeletal muscle because of the dystrophin complex being missing. The vascular smooth muscle isn't signaled properly. And so you've got whole areas of muscle during exercise that are actually ischemic, which also leads to destruction of the muscle. And furthermore, there's calcium handling problems, both in the skeletal muscle and in the heart. And probably in the heart, this is perhaps the most important mechanism that drives the cardiac pathology. So that's basically summarized in this little cartoon, which basically says that, dystrophin has this primary mechanical role. It's involved in transmitting the force that the muscle generates to the extracellular matrix, to the tendons, to the bones, but also it has these other important roles that are lost, including this organizing role with ion channels and the protein, the enzyme that makes nitric oxide to signal the blood flow, as well as a number of other signaling roles. And so if you can go after all of these things with different drugs, then in combination you may have drugs that individually have benefit, but it may not be a huge benefit, but in combination you have the potential to have a major benefit. So I know many of you are familiar with steroids, and most of us think that steroids improve function in the boys because of the fact that it does improve this whole cascade of injury and repair, which in Duchenne patients, the inflammation levels are so high that it creates signals that are not resolved, and this leads to fibrosis of the muscle rather than repair of the muscle in the long term. And so that's why over time the muscle's replaced with connective tissue and then eventually with fatty infiltration. So we think that's why steroids improve things somewhat. But as you'll hear from Nick, and as you probably heard from others, the steroids also have some negative consequences that makes them a much less than perfect drug, and a drug that we'd really like to see replaced by something better that could have a similar impact, a similar positive impact. You can visualize what I'm saying directly using an MR, and that's something that we've been doing in Florida by following about 130 Duchenne patients over time and watching the changes in various muscles. And what you can see, and here's a cross-section through the thigh of different boys at different ages, and what you can see is over time the muscle is gradually replaced by these light areas which represent fat. But using different types of imaging, what you could see is that these early ages, the muscles are highly inflamed, and then they start to become fibrotic, and then after they become fibrotic, you've got infiltration of the fat. So it's a progression that goes all the way from here to here, but the rate of that progression is something that drugs can address, and we think, if you had enough drugs going after these various pathways, you might be able to greatly slow this progression and change the course of the disease altogether, as opposed to steroids, which only slow this down by a couple of years in progression, and as I said, also have negative consequences that work against you as well. So the opportunity here is to go after these various mechanisms, and so the real opportunity is then to combine these, to go after drugs that hit all these different pathways so that in combination they do much more than what any one drug alone can do, and to even combine it with the dystrophin restoration therapies and eventual utrophin restoration therapies that are ongoing in the clinic now. So the first type of therapy, which actually has started in DMD trial, is to try to improve the growth and repair of the muscle by targeting the myostatin pathway. I'm sure you've all heard a lot about the myostatin pathway. Myostatin is a negative regulator of skeletal muscle growth, and so it basically slows the proliferation of the satellite cells. It blocks the hypertrophy of the muscle fibers themselves. If you take myostatin away, if you take it away from birth or before birth in these cattle, you get these giant muscles. The same happens, at least in 2 reported human cases. But the idea is that if you do this in a Duchenne patient, you're not going to get these giant muscles, but you are going to get muscles that probably grow more, become stronger, but more importantly, repair more efficiently, and therefore slow the progression of the disease. And we've seen this both in mouse models of Duchenne as well as in the dog model of Duchenne, as I'll show you in a moment. And so this is all signaling through a receptor that is involved in what is called the whole activin family. So it's an activin receptor. And what this means is there are a variety of ways you could try to inhibit myostatin signaling, all the way from making antibodies against myostatin to overproducing a molecule called follistatin, which you've probably heard has been in gene therapy trials, as well as even using soluble receptor, which was in a failed trial. So all of these approaches have been under development by industry, fortunately for us, And as I said, Pfizer has now launched what is the second, or actually the third trial in Duchenne going after this pathway. But I would say that the fact that the two previous efforts have failed doesn't mean that this is not a good pathway for Duchenne. It just means that the previous trial didn't have reagents that were well suited. So the Myo29 trial that was done by Wyeth, the antibody really just didn't work. So Pfizer developed a new generation antibody that seems to be much, much better, and they've started dosing Duchenne boys with it. The soluble activin 2B receptor that Acceleron had in trial was halted because of bleeding, but that's because it really was hitting too many targets. If you look at what the soluble receptor actually binds, it binds a whole number of molecules that are related to myostatin, but many of them have many other important roles, including this BMP9 and 11, which are involved in vascular development, and therefore probably when you inhibited their signaling, it caused these bleeding problems. So there's every reason to think if you target myostatin directly and don't target all of these other molecules, that it could have tremendous benefit in the Duchenne boys. So that's what we're going to find out in the Pfizer trial. We've done dog and mouse studies, as I said, to look at this. And what the dog studies showed us is something that we hope is modeling much more closely what we might see in humans, because the dog disease is much more severe than the mouse disease. The dog fiber type distribution is much more similar to humans. And also we were able to ask other questions that are relevant to the human disease, including this potential acceleration of the cardiac disease, because that's the one potential downside of myostatin inhibition, that it might have negative consequences for the heart in the long run, but we don't know that at this point. At any rate, we looked at the change in mass in muscles in the dogs, and what we did, the way we did the study, our time zero was actually 1 year of age in the dog. So we waited until these dogs had developed considerable pathology as well as were fully grown, and then added on the myostatin inhibitor to get a more realistic appraisal of what you might see in the boys. And what we saw was quite a bit of improvement in terms of the size of the muscles. They got on the order of 15 to even 25% larger over the next 6 months. We also saw impressively decrease in the overall levels of fibrosis and almost a total stoppage of the fatty infiltration in these muscles. So it didn't get rid of fibrosis, but certainly the situation was much improved. And furthermore, all of the muscles we looked at looked to be improved, and the heart was not any worse. The heart was not any better, but the heart was not any worse. And so we think, at least in these shorter-term studies, we didn't see an acceleration of the cardiac disease, although this is still something we've got to be mindful of, and we need to use cardioprotective drugs in these cases where we're looking at myostatin inhibition. Now, the other thing I want to talk a little bit about is also in trial, and we just got word today that the Lilly trial with tadalafil is fully enrolled for correcting blood flow, but another trial is about to start, hasn't started yet, which is going after this decreasing inflammation and fibrosis. So it's not a trial that you've seen enrolling yet, but the idea is to go after a molecule called NF-kappa B and its downstream signaling pathways, because we think that is the target that gives the beneficial effect of the steroids, that this NF-kappa B signaling, both in the inflammatory cells and in the muscle cells, leads to a lot of the pathology, inhibits the muscle regeneration. So if you could dampen that, it should do quite a bit should give a lot of benefit to the muscles, especially during the hyperinflammatory periods. And so that's been shown in mice, and we've now done experiments in dogs that also bear this out. So I'll just tell you a little bit about what we did in dogs, which was actually using this compound that's going to go into the clinic in 2015, called CAP1004 from a company called Catabasis. And it targets, in particular, this arm of the NF-kappa B signaling that is pro-inflammatory, and also it inhibits the muscle regeneration. And what we've seen is very successful target engagement in the dogs. When we used it in mice, they ran much more on running wheels. It preserved their diaphragm function, but more importantly, we were able to recapitulate this in dogs. We saw less fibrosis in their leg muscles. We saw that their diaphragms were much better maintained, and when we measured their pulmonary function, it was markedly better than when it's not there. And we think that the reason the diaphragms in particular get so much benefit is when we look at both the dogs and the mice, that's one muscle where the inflammation is maintained at a high level for a very long time. Now in the boys, we see that in their leg muscles, it's maintained for a much longer period than in the mice. So we think the dogs are a better model again than the mice, especially looking at something like an NF-kappa B inhibitor, where your main target is going after the inflammatory response. Now, the last thing I'll tell you a little bit about is this issue of correcting blood flow. As I said, we heard today that Lilly just heard that their tadalafil trial is now fully enrolled. This trial is evaluating skeletal muscle function based on the idea that what this drug does is it inhibits PDE5. Now, that's an enzyme that interferes with buildup of cyclic GMP. Cyclic GMP, in turn, is necessary to activate enzymes to phosphorylate a number of targets in the cells, but most importantly, in smooth muscle cells that control the vasculature, when you build up cyclic GMP, you relax the vascular smooth muscle. So you get better blood flow. So that's the whole idea of this. Now, there's been reports in mice that you could also get cardiac benefits, but many of you may know that there was a failed trial of sildenafil looking at cardiac function in DMD boys, which didn't show very much promise. But we think that that's because the boys were probably too far along in their cardiac disease progression. And so we wanted to look at it in dogs again before the cardiac disease started, to look to see if there's both benefit to the skeletal muscle in terms of blood flow, as well as whether or not there might be significant impact in the heart. And just to summarize that data, here's the rationale, is because not only does inhibiting PDE5 and building up cyclic GMP lead to relaxation of the vascular smooth muscle, but it also buildup of cyclic GMP within the muscle cells, within the skeletal muscle cells and the cardiac cells, phosphorylates a number of important targets, including these channels, these so-called TRPC channels, which leak calcium into the cell. And we think this is one of the major problems that the heart has, that these calcium channels are leaky all the time, and the heart has to deal with this recurrent calcium overload. So what we saw in the dogs, first of all, was that, in fact, we are able to phosphorylate these PDE5 channels, and we saw that in the dogs, we saw improved skeletal muscle histology, and we saw this focal necrosis that we think is because of the blood flow problems went away. We still saw fibrosis, but we no longer saw these large clusters of necrotic cells. But the main thing we saw was that the cardiac function was very well preserved, as was the cardiac histology. And so it really greatly delayed the onset of the cardiac disease. So this suggests that in these ambulatory boys that are taking tadalafil, the short-term goal is to demonstrate that their fatigability is improved because their blood flow is better, and that preservation of their muscles in the legs is better over time because they're not having this focal necrosis due to being ischemic. But at the same time, we think that 5 or 10 years later, at a later age than what these boys are being treated, we're going to see that the onset of their cardiac disease is greatly delayed by the use of tadalafil. So that leads us into the idea that Nick is going to talk more about, that we really need to start adding these drugs together. So there are cardiac drugs that are currently being used. Boys are often on ACE inhibitors or so-called ARBs, and more there's a push to add on aldosterone receptor antagonists such as eplerenone. And there's been a trial that's been recently finished, and we'll soon know the results of that. So if you've got that cardiac cocktail and then add on tadalafil, that could provide even much greater benefit to the heart. And of course, there's going to be skeletal muscle benefit by adding on tadalafil as well. Nick's going to talk about tamoxifen, which really is a drug that will target some of these calcium handling abnormalities and maybe through other mechanisms will benefit the muscle. And so that would be another drug that you would want to add on to what the standard cardiac complement would be. And then the list goes on, because as we move beyond these approved drugs, we'll want to then start adding on some of these drugs that are in the clinic as they become approved. And then a number of candidates to look at, including the myostatin inhibitor that's already started in trial, and this CAP 1004, which will start next year. So that's basically where we are. That's the rationale for doing this. I mean, in a sense, I always like to use the AIDS example. Magic Johnson still has AIDS. He's never been cured of AIDS, but he's got a cocktail of drugs on board which has essentially arrested the progression of the disease. That's our goal for Duchenne, and we've got to start doing this first one drug at a time and then start getting them together in the kids in combinations after we first see which combinations are safe and effective in our best animal models. And with that, I'll stop and then turn this over to Nick.
Okay, Nick.
Okay, well, thank you, Lee. I'm actually not at University College London. That's a separate institution. I'm at the Royal Veterinary College in London, and one of the reasons I'm at the Royal Veterinary College is that we are raising a novel colony of dogs, which we think will be useful in much the same way Lee's talked about. But what I'm going to talk about today is mostly concerned with mice. I'm going to talk really just to back up Lee's comment about the case for combination therapies. I'm then going to suggest a standardized approach to testing those combinations, which we're working with and I think has some advantages. I'm going to give some examples of work that we've done that hasn't yet been published, although it's in preparation, and then talk about possible combinations for the future. And a lot of this will echo what Lee has said, but I'm finally going to add with a few cautions of things that we've got to think about quite carefully as we go down this pathway. So, as Lee's pointed out, even if we replace dystrophin, we've still got to deal with the existing pathology, and it looks like the methods of replacing dystrophin are not only mutation-specific, but they're unlikely to be 100% efficient. And even if we go down a gene therapy route and putting in a modified version of dystrophin, it's unlikely that it will be the magic bullet that will solve all the problems. And so for downstream pathology, there are a multiple set of targets. And the advantage about treating the downstream pathology is, of course, that will be applicable to all DMD patients and not specific mutations. Essentially, we are already doing combination therapies in our clinical trials because the widespread use of corticosteroids means that most of the drugs that we're testing in clinical trial, the new drugs, are actually being tested in combination with corticosteroids and also in combination with different cardiac medications. I'm focusing particularly on repurposing existing drugs because that shortens the time to get them into the clinic and get them used in patients, and we have a lot of advantages. We already know the toxicology, we already know the side effects, so we can predict some of the problems that might happen. This slide, it shows essentially pretty much mirrors again some of the points that Lee was making, in that the absence of dystrophin doesn't just lead to a problem with instability of the muscle fibers, which is this activity-induced damage, but it also causes a loss of cell signaling. And as Lee had pointed out, the influx of calcium causes a lot of dysregulation, leading to hypercontraction of the muscle, problems with the mitochondria, activation of internal proteases. But a combination of all those things going wrong in the muscle, together with external inflammation in response to trying to mop up the damaged muscle, leads to a lot of oxidative stress, nitrosylative stress. So there's a lot of things going on within the muscle fibers that are causing modifications of their function. And one of the effects of losing the signaling and having these various stresses is that you actually see a loss of strength, a loss of force generating potential within the muscle. And that's quite an important element. So not only are the muscles weak, smaller and damaged, but they're actually per unit muscle much less strong as well. And of course, all the inflammation and fibrosis leads to a failure to regenerate muscle properly, and it becomes a very vicious circle. So there's a whole range of targets. We'd obviously like to increase the stability of the muscle membrane, and although we think that dystrophin and utrophin are the two best candidates to do that, there may be other drug treatments that can do that. We'd like to be able to increase muscle strength, and the myostatin is the classic example, but there may be other ways of addressing that as well, particularly in generating more strength per unit muscle. Clearly, we'd like to reduce the inflammation, which should in turn reduce the fibrosis, but there may also be room for drugs that specifically target parts of the fibrotic process itself. We'd like to increase the blood supply because the poor blood supply, particularly in exercise, is undoubtedly part of the pathological cascade. Need to be able to improve the regeneration of the muscle, improve the ability to deal with the excessive calcium, and the list goes on and on. So we've looked at a lot of what's been published and tried to think of a way of having a standardized approach to screening different compounds in a way that we can really relate all of those different effects, and we can look at additive effects and make what I hope are logical thoughts about what drugs should be added. So we think it's very important to use MDX mice with established disease. And by that I mean not a disease that has started. So in the MDX mouse, there's no disease until about 3 weeks old. And then there's a very rapid inflammation, damage to the muscle. But that's still essentially the very, very early stages of muscular dystrophy, and is equivalent to a neonatal boy with DMD. So really, looking in that area is really equivalent in terms of the pathology to what's going on in a newborn. And at the moment, that's not our primary population that we will be addressing. We're addressing patients who have established disease. And for that reason, we use older mice. Even though the disease is not as florid, is not as obviously very inflamed in the older mice, it is still established disease rather than developing disease. We dose for long enough that we will see the full effects and potentially the side effects, and so we do that for 12 weeks. And a short-term test of a few weeks doesn't really tell us of the potential for a drug that we may be suggesting patients are going to take for the rest of their lives. We think, very importantly, the primary outcome measure when evaluating a therapy should be one that reflects the goal or goals of the therapy and is not subject to mouse motivational state. One could think of, for instance, mice being given drugs that affected the central nervous system that might cause them to be more active, but that actually doesn't mean that that's a good treatment for muscular dystrophy. And obviously there's a number of secondary outcome measures one can think about, including histopathology and various other things. It's very important also, and Lee's already touched on this, the choice of the muscles that we look at. And finally, if we're trying to think about using our animal models to give us information about what we should do for human trial, then it's useful to try and look at things like serum biomarkers and MRI, as Lee's commented on, as these are the sorts of things that you can look at in patient populations. So we focused a lot on measuring the muscle physiology, that is the ability of the muscle to generate force, and most importantly, the ability of the muscle to withstand the effects of exercise. And an exercise that's particularly damaging to dystrophic muscle is eccentric exercise. And that's called it's a lengthening contraction. So normally you do a concentric exercise when, say, you're lifting a weight. But if you lift a weight that's too heavy for you, and despite all your effort, you're starting to drop the weight, then that's lengthening your muscle, and that's particularly damaging. And you see that when walking, trying to balance the effects of gravity. So walking downhill involves a degree of eccentric exercise. And it turns out with the lack of dystrophin, your muscles are much more vulnerable to this sort of exercise. So that's a very important measure that we use. And if we can decrease the damage associated with that exercise, then we think that's a very good indicator of the ability to reduce the amount of exercise-induced damage in the muscle. And obviously, if we could stabilize the disease, in other words, prevent exercise-induced damage to the muscle, then we'd like to increase the force that that muscle can generate. And that's really what we're thinking about there, is the amount of force per unit quantity of muscle. Although you could also think about the amount of muscle, and that's really what is the target of the myostatin-based therapies. So we use two approaches, either testing the muscle as it's sitting in an anesthetized mouse, or taking the muscle out and testing it in an in vitro system, and focusing on two muscles, the anterior tibial muscle for the in situ and the diaphragm muscle for the in vitro. Because looking at those muscles like that, there's no effect to the motivational state of the mouse compared to, say, trying to run the mice on a treadmill. And this shows the sorts of things we look at. So on the graph A, we've got specific force, so force per unit amount of muscle on the vertical axis, and the frequency of stimulation on the horizontal axis. And the top trace is the wild-type mouse, and the lower trace is the MDX mouse. And you can see the MDX mouse is very much worse at generating force per unit of muscle mass. And if we can do something to that mouse that brings it closer to the wild-type mouse, then we think that that will be an important way of increasing strength. In panel B, what we've got is this response to eccentric exercise. And what we're looking at here is you stimulate the muscle, make it contract, and then you lengthen it by about 10% of its normal length. In the wild-type mouse, that has absolutely no effect, and that's the top trace on graph B. But the lower trace is the MDX mouse that loses about 60% of its force-generating capacity after just 10 of these eccentric stretches. And if we can change that MDX mouse characteristic up towards the wild-type mouse pattern, then that tells us that we're probably able to block the injury caused by normal day-to-day exercise. And then, as I said, we also look at the diaphragm, but the diaphragm responds very differently to a number of things compared to the limb muscle, so it's worth doing both. You're now seeing a slide that looks at the limb muscle, the tibialis anterior or tibialis cranialis, which is the muscle at the front of your lower leg. And we've got, again, on the vertical axis, the specific force, and on the horizontal axis, the frequency. The black line shows the wild-type pattern. The red line shows the MDX pattern, in this case labeled control. And in this particular experiment, we've treated the mice with prednisolone, or tamoxifen and prednisolone. And the blue line is prednisolone, and that really makes no difference to the force-generating capacity. But tamoxifen definitely does increase the force-generating capacity, and the combination of tamoxifen and prednisolone substantially increased the force-generating capacity. So here's a beautiful example of where putting two drugs together has an additive effect, and therefore really shows that we've got a very promising drug approach that addresses the ability to generate force. However, the next slide is the eccentric exercise experiment on the same treated mice. And again, black is the wild type, red is the MDX. And here you can see that unfortunately tamoxifen does not affect the resistance to that eccentric exercise. So whilst it's good at helping to increase muscle force, and therefore is clearly affecting one part of the pathway, it unfortunately doesn't do anything to address the deficit caused by exercise. You'll notice that prednisolone partly addresses this, and this is probably one of the reasons why prednisolone slows down the progression of the disease in DMD. So on its own, or even with prednisolone, tamoxifen is not going to be particularly helpful just as a drug, either on its own or combined with prednisolone. So we need to look for something else. here we have another drug, and again, this is an approved drug for use in humans. And this is, again, the eccentric exercise graph, because there was no effect of this drug on the force-generating capacity. So we don't, in this case, have a wild-type mouse on this trace, because it would make it complicated to look at. But the circles, which circles and triangles fall on pretty much the same line, the circles are the MDX mouse, and in two different concentrations of this drug, one of them does no good at all, and it's actually the higher concentration, and one gives us a very significant reduction in the amount of force loss associated with eccentric exercise. So just from those simple experiments, one could see that potentially a combination of tamoxifen plus this drug A plus or minus prednisolone has the ability to address both elements of muscle physiology, an increase in force and a resistance to exercise-induced damage. And because these are already approved for use in man for other indications, this potentially is something that we could take through to clinical trial quite rapidly. Although we do need to consider that our initial assessment in mice needs to be backed up potentially by an assessment in dogs or by careful toxicology to make sure that there are no adverse consequences of those interactions when we get to man. So I've focused so far on muscle physiology, but I just want to mention that histopathology tells us a lot, because we can look at changes in the fibrosis in the muscle, variation in fiber size, which is typical of a dystrophic process, how much inflammation there is, and we can look at it either in standard histological stains or using specific antibody stains. And I'm just going to show you with another therapy, it's actually one that restores dystrophin expression, what the effect of that is. So here we have the anterior tibial on the top left, and the no treatment in the red bars shows, this is frequency on the vertical axis and this fiber sizes on the horizontal axis. And you can see in the untreated MDX mouse, there's a lot of very small fibers typical of the dystrophic process. But when we restore dystrophin over a long enough period, we get a much more normal distribution that you see with the black bars, telling us that we've gone much more back towards the wild type state. However, exactly the same mice treated this way to have that effect on the TA, we see a very different effect in the diaphragm, where we really don't see a normalization of fiber size, and we see some very large fibers, which you can hardly see on this graph at this magnification, in the 35 to 50 range of fiber size. And that's reflected when we look at the variation in fiber size, which is on the lower left for the TA and the lower right for the diaphragm. And for the TA, the effect of treatment restoring dystrophin reduces the variation, as one might predict. But it's interesting, in the diaphragm, it doesn't. It actually increases the variation because we're generating these much larger fibers as a response to treatment. And this shows, I think, very vividly why you need to consider different muscles and look at several. I think probably I'm going to skip this one, but basically what this is about is that we can, particularly with the TA, find histological explanations for changes that we see in muscle physiology. The reasons that we do this standardized assessment is that allows us to compare single drugs and combinations of drugs, and potentially the same methodology can be reproduced in other labs. And so the whole reason for looking at muscle physiology as the primary outcome measure is we think that that is really focused on the translational potential to the clinic. The slide here of further combination therapy examples pretty much says what Lee's already said. So by genetic therapy, I mean an exon skipping, or microdystrophin gene therapy, read-through drugs, or potentially the upregulation of utrophin, which you might then want to combine with something that makes the muscles larger, such as myostatin inhibition, or something that increases the blood flow, such as the use of tadalafil, or an antifibrotic to try and reduce the existing fibrosis that's there, or maybe using NF-kappa beta inhibitor as an alternative to the use of steroids, combined again with similar sorts of drugs. And you could write pretty much an endless list of things here. But we need to be careful. First thing is, whenever we're thinking about using animal models of human disease, we need to be aware that positive effects seen particularly in the mouse may not be seen in DMD. And one of the things we can do in order to reduce that risk is to keep the doses that we test in the mouse within the normal therapeutic range in man. This is particularly true when we're looking at drugs that are already approved. And a lot of the translational problems with using the mouse is tend to use very high doses, sometimes by the wrong route of delivery. Side effects of combinations may not be seen in the mouse and may be seen in man, so we need to be very much aware of that. We need to be aware that many of the drugs that we might look at for repurposing may not have been previously used in children, and children can respond very differently to a drug that is safe in adults, and I think that's a very important consideration. We do, however, know that, for instance, the case of tamoxifen, it has been used in teenagers, actually, interestingly, in the treatment of certain cancers. If we start to think about doing combinations with currently unapproved drugs, so experimental drugs, then that brings a very significant regulatory burden in terms of the toxicology and safety pharmacology. So I think the point Lee made about when drugs are approved for use, say, in DMD, we need to think about combinations of those drugs with other drugs. The problem we have in assessing that before they become approved is that requires collaboration with or between companies. And if you're a company trying to develop a drug, what you don't want to do is have people use that drug in a way that might potentially generate adverse events, together with obviously wanting to safeguard your intellectual property. So I think the ones that we're going to be able to assess as combination therapies in the next year or two are going to be those drugs that are currently approved for use in man, but as I hope I've demonstrated, have some real potential to modify the disease process, at least in the MDX mouse. And I think with that, I'll stop and we'll open it for questions. Thank you very much.
Okay. Thank you so much, Dr. Sweeney and Dr. Wells. I want to remind people that if you have a question, you can type your question into the message box, and we will see it, all of the presenters. And I just want to add a little explanation for why we're talking about combination therapeutics now, beyond the obvious that it looks like it's very much the direction that we need to be taking therapies for this disease. You've probably seen our holiday campaign appeal. PPMD is trying to put together a pot of money, because in January we will put out a request for application, and we will ask investigators from all over the world to submit an application that to try these drugs in combination and to explain what they want to try and why and how they're going to do it. And then they will all be reviewed very intensively. So it's a bit of a contest, and we will fund the top scoring applications. So that's what we're going to be doing with the holiday campaign funds. That's part of the fund. There will also be funds left over to work on the regulatory aspects. As Nick just explained, it's more complicated to get regulatory approval when you're using drugs in combination. And that's just due to the confusion of not knowing sometimes which drug might be causing a certain side effect, or whether it's caused only by the combination of the drug. So we want to start working on these things now. We don't want to be in a position of having a couple of therapies that are approved that look promising and could be useful potentially together, and not being organized enough to do that as soon as possible. So this is laying the groundwork. And I'll just start off as questions are coming in and ask both of you to, I think what people really want to know, what people have been asking me, is what two things would you try first, or maybe what three things? I think you both hinted at some different possibilities, but I think people would be curious to know what you would think would be at the top of the list and why.
That's a really good question. Does Lee want to start off?
Yeah, well, so, what we're planning to do right now is I want to add tadalafil, especially since a year from now, that trial will be done, and we'll know whether it impacted the skeletal muscle in a big way. I want to do some preclinical studies in dogs where we put tadalafil on various combinations of the cardioprotective drugs that are currently being used, and then see if there is significant added benefit, but also if there's any downside, as Nick has alluded to. And also, of course, at the same time, we'll be looking to see what the benefit is to the skeletal muscle when you've got this combination of drugs that are primarily targeting the cardiac disease. So the other one that the other approved drug that Nick has worked on quite a bit and we think really deserves some careful dog studies is tamoxifen, because it does look like it's got a fair amount of potential, and so we'll be layering it on to these combinations of drugs that are really designed, more for the heart at the same time. Now, we also are going to do arms with and without prednisone because, I think prednisone clearly can show benefits and clearly does show benefits, but the question is, at some point, is prednisone going to create problems for some of these other drugs? And those of you who saw the recent paper from the Ohio State group basically showed, at least in the mice, that if you layer prednisone on top of eplerenone, it actually blocked the benefit of eplerenone. So we need to do these dog studies where we've got a better model in terms of what prednisone is doing, because we've got much more information. We need to do a lot of these studies in dogs with and without prednisone to know when it's going to actually be masking other benefits, and if there's a point where we can actually withdraw the prednisone, because I'm sure most of you would rather never hear about prednisone or prednisolone ever again. Now, I personally think that is a goal that we can achieve, especially if some of these other drugs have some of the major anti-inflammatory actions that prednisone does without some of the other targets that prednisone hits. So at any rate, that's a long-winded way of saying that, we're going to focus really on tadalafil and tamoxifen early in combination with the approved battery of cardiac drugs, because, we think that's the fastest way to change the management of these patients. And obviously, as these other drugs get approved, if the myostatin inhibitor gets approved, if the NF-kappa B inhibitor gets approved, then we want to have studies ongoing so that they're nearly completed, where we've actually modeled those drugs on top of the best approved drug cocktail, so that we know, basically, if you go to using 3, 4, 5 different drugs, is there a major problem with doing that, or is it all major upside? So that's what we plan to do in the next year or so, while we're waiting for some of these new drugs to finish their trials.
Okay.
And my answer is going to be similar to Lee's, although I'm more focused on the sort of physiological effects in the skeletal muscles. And one of the points I'd like to make about that is that I think that this is potentially applicable to a wide range of patient ages. If we can stop progression of the muscle loss, the muscle damage and muscle loss, and increase the ability of the muscles to generate force in a boy who's in a wheelchair, that will mean that the arms will be very much more effective. Hopefully the respiratory muscles will be more effective. And so that will have a major impact on quality of life. So this is not thinking about therapies that are designed for a very narrow window for ambulant children only. No, we're thinking much broader than that, I think, when we're doing these.
Yeah, I would echo.
Yeah, sorry.
I would say, I would echo that. I mean, we basically are looking at this as 3 targets: the heart, the diaphragm, and the limb muscles, because the drugs will interact with them differently, as Nick's already pointed out. And so we evaluate them separately and are hoping for combinations that improve all of them.
Absolutely. And then my particular tack on this is I've given you an example of a drug that seems to improve force and a drug that seems to improve the resistance to eccentric exercise. So if we can couple those two and get both of those benefits, then we're a good way towards essentially a pharmaceutical version of what dystrophin's doing. And it may be that gives us at least a drug combination that achieves most of what we're trying to achieve with restoration of dystrophin. And so that's our particular focus for the next year or two. Obviously, tadalafil will be really important to put into that, and we're certainly looking at tadalafil with 1-2 other combinations, and plan to do a lot more. Although in our case, that's very much subject to funding. Probably the case for Lee as well.
Well, we'll have an RFA coming out if it happens. So we're going to go through some of the questions that have come in while we're talking, and someone is interested in better understanding the side effects of tamoxifen.
So we've looked at that in terms of what's in the literature, and the side effect that worries me the most is the transient cognitive impairment that's been reported for a number of people. The group that's been most widely treated with tamoxifen is women who have been treated for estrogen-responsive breast cancer, where tamoxifen appears to be very, very effective. And we get the literature is full of reports of people who either had this cognitive impairment, this fogginess in their thinking for the whole time they're on the drug, others for the initial sort of few weeks on the drugs, and others who never have this effect. So it's those sorts of side effects that we can look at and predict what the potential problems are and what to look for. We've looked very carefully in the mice at, for instance, the effect of tamoxifen treatment on spermatogenesis, testis size, those sorts of things. And at the moment, it's not looking like that's going to be a serious side effect, but we clearly don't know because there's not been enough children, especially pre-pubertal children, who have been treated with a drug like tamoxifen, and potentially that's another possible side effect. So the advantage of the approved drugs is we know quite a lot about what they do and therefore can predict what the possible side effects are and what we would need to look for and what we need to focus on in the preclinical studies in the animal models.
Okay, thanks. And someone else has asked about using over-the-counter medications, in this case, such as idebenone, CoQ10, or creatine in combination with maybe things that are, require prescription.
I'm happy to start off with that and then pass it over to Lee. Where trials have been done with those sorts of over-the-counter medications, it doesn't appear that they have a very obvious beneficial effect. And a lot of work by the Synergy Research Group looked at those sorts of drugs to see whether they could see a beneficial effect and really couldn't show that convincingly. In the mice, some of those have looked quite promising, but when you look at the doses that the mice were given and you scale those doses appropriately for human, it would mean consuming very, very large quantities. We know with many of those things, such as creatine, if you take too much of it, you have problems with diarrhea and various other side effects that really would be unacceptable. So that's the point I was making about in our preclinical studies, we have to dose at a dose that is translatable into human medicine and not a dose that causes the best effect in the mouse or the dog, because we're not looking for the best effect, we're looking for a translatable effect.
So would it be fair to say...
If the questioner's point was, is that going to interfere with this, I think most of the companies that have taken the drugs into trial are just looking at it that if you're on a nutritional supplement, be it CoQ10 or creatine, then they would prefer you, they're asking you just to be stable on it and not go off of it. I think most of them feel like Nick does, that the impact is somewhat minor if you can show it at all, and therefore it's not likely to interfere with their drug approval process. But they still prefer, for all supplements, people just not change them when they go into the trials.
Right, and I think, the questioner may have also been wondering if those things are combined. If you combine some of those OTC things, do you see a greater effect? And
well, well, it's certainly possible that some of these things that show some minor benefit in mice might also show minor benefit in people if you had other things that were giving you much more major benefit. So it is possible that even though as a monotherapy you can see nothing, that it could give some incremental benefit if layered onto much more effective therapies.
Right. And,
yes,
we actually
have
data. I was going to say, we have a little bit of data from Duchenne Connect, the registry that PPMD maintains that has about 3,000 records. People record what types of over-the-counter medications they're giving. And there's enough data in there now that we can start to tease out whether or not maybe that's having an effect, with lots of caveats, meaning that this is not perfect data. This is, there's a very flawed data set, but when you get enough of it, you start to see hints. So we're actually tracking that information for people who are registered in Duchenne Connect. Because this experiment is being done all over the United States. I dare say most of your children are probably on some type of cocktail supplements now. It's just not uniform enough in most cases for us to be able to draw conclusions about it. I'm going to ask one final question that is a very good one, and that's what is the proposed plan to accelerate the approval or use and approval and testing of combination therapies? And I think what you're seeing right now is the beginning of that plan, and that is that we want to be prepared in advance. We don't want to be in a situation where we have drugs that really look like they're very promising monotherapies, and we're not prepared to test them in humans because we haven't done the background work that we need to do. And also, part of the funding that we're going to use from our holiday appeal is really to look at the regulatory pathway. There is a guidance from the FDA about combination therapeutics that maybe isn't perfect for our uses, and we're quite vocal about going to the FDA and letting them know where we think there need to be improvements or changes. So that's part of what our funding is going to do, is really work on a regulatory strategy. And I have sort of a question and follow-up to Dr. Sweeney and Dr. Wells, and that is, what do you think the potential is for getting a combination therapeutic approved as a combination, so not as separate therapies, but approved for the first time for use in combination?
Well, I mean, if a sponsor has, the tox on them separately and together, then the FDA is open to a sponsor bringing forward a combination and not having to get them approved separately. But then the sponsor has to have control over both entities in order to do so.
Mm-hmm. Right. So I think the challenge is the same sponsor would have to have the compounds in hand.
Yeah, I mean, I guess it would work if one of the compounds was an approved drug and it was off patent, and therefore they're repurposing it.
Right. Okay. So let's follow up with one final question, and that is about conducting appropriately powered clinical trials of the combination therapeutics. And the idea is that to try drugs in combination, you might actually need more people in a trial than you would to try single drugs to be able to tease out what's really happening. And are we concerned about our ability to recruit for trials? That's actually a big question. I think it's really relevant to recruiting for any trial right now.
Yes, it is. And the problem at the moment is that essentially everybody is focused on a very specific age group, about 7 to 9 year old, where you can see a very marked effect on the 6-minute walk test. And that particular patient population is being targeted very heavily. And I think what we need to think about is trying to look at other functional tests which the FDA will accept as tests that indicate or are indicative of potential of clinical benefit. And there are now a number of assessment tools, for instance, that are being generated for non-ambulant DMD boys. And that's a group that currently is not being recruited to trials. And I think we need to be more inventive about our trial design, because we can't just have everybody competing for that very specific group that shows a reliable decline in the 6-minute walk distance.
Right. I think that's a hot topic of conversation now, and then, there's a really active effort underway in many different areas to develop new endpoints or endpoints that can better capture a range of ages. And progress is being made there. I can tell you that we need enough data to be able to validate those outcomes, but that's happening. So hopefully soon we'll start seeing a wider range of ages used in trials. So with that, I want to thank both of you very much. I appreciate it, particularly Dr. Wells, because it's quite late in the UK right now, for doing this webinar and giving us your ideas, and it's very thought-provoking. So I think we're moving into a new era of therapy for Duchenne. Hopefully we will have good treatments, single treatments soon, and in combination we might really start to have very significant impact on the disease. And we are all hoping for the scenario that was achieved with AIDS, which went from an epidemic to a manageable disease. We really want to make this disease manageable and stop the progression, ideally. So thank you all for tuning in.
Yeah, Sharon, I would just say I see there are a few questions we didn't get to because of time. If people want to email them, then we can try to answer them by email.
Right,
Absolutely.
That's true. There are a couple right at the end. I'm sorry about that. We will try to follow up for sure with Dr. Sweeney and Dr. Wells, and we'll make sure you get those answers. Okay, thank you very much. Thank you all for participating, and have a good holiday.
Thanks for everyone.
Thank you. Bye-bye.
Bye.